There we go. Recording has started. I'm going to be posting this over the weekend, so you should for sure be able to see the recordings by Monday afternoon. And I hope you can join us next week. Next week, we're going to hear Dr. Chandra Arora from LA Mission College talk about project-based learning with adults in her classroom, and Dr. Susan Ingersoll from Valencia College will talk about, oh wow, she's here. I'll admit her right now. She's going to be talking about, oh gosh, I can't remember, BioSpark, which is her program with developing all kinds of internships and making all kinds of industry connections, because she's been doing some wonderful things too. Let's see, is there anything else I have to say here? All right, so just so you know, today we will have the two talks will be 20 minutes. We'll do them back to back, and then we'll have questions. And after that, we are going to have a survey, because we can't let you attend anything without making you fill out a survey. But it's really helpful for us. It not only does it let our funder know that these are valuable events, it helps us tune what we do and make sure that we make these the most valuable things that we can for you, because we know you're all crazy busy and always short of time. All right, so without further ado, I'd like to introduce Dr. Misty Wehling and her team to talk about implementing course-based undergraduate research in a community college in Lincoln, Nebraska. Take it away, Misty.
Well, thank you all for joining us today. We're just going to introduce ourselves a little bit as we get started. My name is Misty Wehling, and I teach biology and some biotechnology at Southeast Community College in Nebraska. I'm actually in a smaller town where one of our other campuses is called Beatrice, Nebraska. And as you'll hear, we've been working with biotech at Southeast for several years, and this is our first ATE project. So Tracy, you want to introduce yourself?
Yeah, my name is Tracy Niday, and I teach on our main Lincoln campus. I teach a lot of the biotechnology classes here.
And I'm Gabrielle Johnson. I also teach on the Lincoln campus. I actually teach our majors biology course, and I'm the co-chair of the Lincoln Science Department.
Okay. So Southeast Community College does get a name because we are in Southeast Nebraska. There are other colleges with similar names, and we do have the urban campus in Lincoln, as well as two more rural campuses, Beatrice and then another small town called Milford. And our service area does cover these 15 counties that are in Southeast Nebraska. And one of the goals of this project has been to really try and reach the more rural areas, especially when we're working with high schools, which for me has meant a lot of driving. So, and we are fortunate also to have hired a project coordinator and is helping to meet those needs as well. So our service area also has many biotech companies that we started working with many years ago, starting with some meetings with industry that led to some internal grant funding that really allowed us to get to know what other community colleges were doing to teach biotech. And we went to visit Betsy in St. Louis as part of that, and also the program in Austin Community College. And that did allow us to get connected with other community colleges and have access to the resources like Innovate Bio offers. And that led to us being able to then offer biotechnology classes and hire Tracy to teach them. But they were in a separate part of our academic transfer program that was a concentration. So there wasn't an actual degree awarded in biotech or certificate or anything. And we really wanted to get to that point so that we could have some more resources to be able to market and be able to tell students that it's going to say something on their diploma. And so along that process, we applied to the MentorLinks program, where Bridget Kirkpatrick from Collin College was our mentor, and she really helped facilitate getting over the remaining hurdles to get our program solidified, and we'll talk about how that works a little bit. And in the meantime, we've also then been able to get some funding from Innovate Bio to implement some course-based research experiences that then led to us being able to work with industry, because that grant had a meeting where an industry partner was supposed to be there with you and a member from your college administrative team. So that really helped facilitate getting things done to move forward and identify a project that could be a partnership. And from there, we offered our first summer workshop in cooperation with our industry partner and realized the needs of the teachers. That led to then applying for another grant that would provide some more equipment at the college level, and we could also potentially use at high schools. And then finally applying for our ATE project that really brings together all these things. And that was funded at the end of June with a start date of July 1st. So we've been going for a few months and been making some progress and finding some challenges along the way. So at SEC, we do have 3 stackable credentials, a 2-semester certificate, 3-semester diploma, and a 4-semester Associate of Science in Biotechnology. And the goal is, of course, that all of those will give skills that allow students to enter the workforce. We've also been trying to be super intentional about making sure all of those classes transfer, trying to develop articulation agreements with other four-year institutions in the area. And then there's a couple of extra components. One is that sometimes students with bachelor's degrees end up coming back, brushing up on their skills and getting the certificate before entering the workforce. We also have a dual credit pathway that's at a career academy that's attached to our main campus in Lincoln for high school juniors and seniors, where they can complete this certificate in biotech by the time that they're done with that program. And I did post the link there if you want to find out more about specific classes and all of that. So the overall goals of this project is to increase awareness of biotechnology at that secondary level, as well as offer then course-based research experiences to our college students as well. And the overall goal in the long term is that then more students will pursue biotechnology as a career. We do have, this grant encompasses 2 projects. The DNA barcoding is for being incorporated in our majors biology, and this has been done in cooperation with the resources available from the DNA Learning Center. And the Canine project is the one that we'll focus on today. And this one is involved high school ag and science teachers, as well as our non-majors biology at the college level.
Okay, thank you, Misty. So I will talk a little bit about this Canine project specifically. So as I feel like this audience, we're all aware that if you can personalize an experience for students in your courses, it really, really helps with increasing engagement in STEM, belonging in STEM, all of these wonderful things that we've been working on. So our project team really wanted to create an experience for students that was integrated into the course and really help them to learn these various biotechnology skills with something that can be personally interesting to them. So the research team found a paper that we have cited at the bottom of the slide with researchers out of Princeton and Oregon State, where they were able to identify 3 gene inserts on chromosome 6 in dogs that are associated with this hypersocial behavior, so really social behavioral traits. So this is something that we thought would be an excellent opportunity to integrate into our course for our students, because many people love dogs. You might have a dog at home, right? It's something very relatable. So we can look at these hypersocial behaviors in dogs. Students are given a phenotype survey, essentially, where they determine what we call our hypersociability score. And then through these molecular lab processes, they are able to determine if their phenotype score matches up with the genotype they're seeing with these gene inserts for these dogs. So once again, we're looking at this kind of hypersocial behavior. Tracy will talk more about the specific gene loci we're looking at later on. But we thought this was a really exciting opportunity to, once again, give students kind of a relatable opportunity that they can then work in the lab and kind of figure out if there's this correlation between genotype and phenotype. And this project also has some really exciting potential broader impacts. As Tracy will mention in a little bit, part of our project involves developing a database that will have available nationally, so students and teachers can put their data into the database and will kind of see information about this project in real time. This could be used for testing eventually for like service dogs, dogs in shelters to see if they will be a good fit for families, just kind of general animal care at home. So a lot of really exciting potential applications and broader impacts of our project. To talk a little bit about our workflow here. So students get to essentially start the project from start to finish, which is also something that we're really excited about for both our high school and college non-majors biology students. So we have an intro lab where they learn how to micropipette. Many of these students will not have had this opportunity previously. They then get to find a dog, whether it's their family dog, their neighbor's dog, a dog in our department. These images on the bottom left here have some dogs from our science department faculty. They get to collect a dog saliva sample using these saliva swabs. They get to extract the DNA in our lab. They then get to run PCR to amplify the DNA, right? And then run their gel electrophoresis. Something we've been really excited about is that we're using these mini-1 systems, which have really optimized things in our labs, right? We probably all know the old school way that's considerably more labor intensive. These mini-1 gels have been awesome because students can put their smartphones on, take the picture, right? They can see the gel running in real time, which has been really exciting in our classroom. They then get to look at their bands of their gel and figure out the genotype for the dog they collected the sample from, and once again see if there appears to be a link between the genotype and the phenotype from their survey. So it's really exciting. They're getting to kind of go through this whole molecular workflow that's really common in biotechnology labs, so they could use this for all sorts of other applications, and they get to actually analyze real data. So this has been a really exciting opportunity.
Awesome. Thank you, Gabby. So one thing I want to talk about is throughout this whole project, I think that the partners have been key for us in helping us to get it started and then also now implementing it in the high schools. So one of our main partners is Neogen. They are an animal genomics company here in Lincoln, and they are who kind of inspired us and pointed us towards this research to create the project. They also helped us by hosting the summer workshops along with Lincoln STEM ecosystems. And it's actually, I mean, they've sort of gone above and beyond. They send some of their employees to our high school labs when we implement this to help out, because there's a lot of students, 24 to 25 students. And not only that, but they share, usually they have employees that come that are probably in their, I would say 20s, early 20s. And while the gel is running, they share their experience about working at the company. And we hope that this really impacts the high school students and gives them an example of someone who they see as a peer doing a career in our, a local career that maybe they haven't heard of before. So I think that's been really impactful. Another thing that we thought about with this ATE grant is this grant is 3 years long. We are providing support by giving the high school teachers equipment and reagents. And then in year 1, we're going into the classroom and actually helping them run it for the first time. But our thought was, okay, after 3 years, when this grant is done, what is going to make this be sustainable and continue on in our community after we're done? So EPSCOR is a NSF funded program at University of Nebraska of Lincoln. They already have a system where they have several mobile biotechnology labs that they supply to teachers, high school teachers throughout Nebraska. So they have kindly and generously agreed to, after the 3 years are over, they're actually going to take our project and develop it into a mobile lab and continue providing the teachers the reagents and the equipment, which as you guys might know, like it's very a lot of this equipment is pretty expensive. It's hard for high school teachers to buy. They also maybe don't have time to prepare the reagents. And so EPSCoR will do that. They've also agreed to host our website, which will have a database where students can, high school teachers and our college students can enter data. And it will, as Gabby mentioned, we hope that in real time it will display an infographic of all of the collected data that teachers can use in their classroom too, to, you know, say, this is your class sample size, this is what our data says, but this is a very small sample size and it may not be reliable. What does 500 dogs look like? So we're hoping that in the future that will be really exciting. And then also Boedecker, who is in the Bay Area, who has been doing this for a long time, they are working with us to help us optimize our protocols so that they're very high school teacher and high school student friendly. So yeah, so the partners are super helpful in getting this going. Cool. And so just an update of what we've done so far with this canine project. We implemented it this fall in all of our non-majors biology classes. We have 8 lab sections. I do run this in the biotechnology 1 classes. I do all 3 gene markers in the biotech 1 classes, and we just do 1 gene marker in the non-majors classes. So it's a little bit more difficulty for the biotech 1 classes. And then we had 7 teachers attend a summer workshop for this last summer. 5 of them were high school teachers. We even had some middle school teachers. And we have started to run those projects in their classrooms. And of those high school teachers, actually 4 of them are ag teachers in rural communities. So it's been really cool to go out into these rural communities, as Misty said, and run these projects with students who may not have had these opportunities before. And then lastly, we're just working on optimizing the curriculum with Boedecker, as I mentioned before. We're also hoping, in addition to the database, we want to make, we are going to be producing a video that hopefully will introduce the project in a more like engaging way for students. And there have been some like growing pains, some challenges as we've implemented this this semester. So we have had a little bit of pushback with our Gen Bio teachers. There's that same old argument of, if you implement this biotechnology lab over 2 or 3 labs, the students might be missing some core concepts. So we're working with our biology faculty to kind of integrate those concepts into our canine project so that it's a solution kind of for both of us. And then this is our first year doing this project. So this first year we're going into all the high school classrooms and we are essentially supporting them and running the project. And then after year 1, those teachers are going to continue to run the project, but they're doing it themselves. So we're kind of anticipating that transition to be a little bit interesting because they're going to be responsible for running it instead of us. And then just getting those supportive materials like the database and the video up and running. So those are kind of the things that we're working on right now. And then this is just some sample data so you can kind of see what the students see. If you look on the right there, this is a gel example. So here is all of the 3 gene loci, the 6.66, 6.7, and 6.6 are their names. And you can see there's 3 possible genotypes that a dog could have. So a dog could have no inserted DNA at either chromosome, so that each dog has 2 chromosomes. And we call that genotype a homozygous wild type. So they have no extra inserted DNA. And according to the hypothesis of that research paper, they should have less social behavior. But again, we don't know for sure. And then the second genotype is where they have inserted DNA. So you can see in that lane 6.7, there's only 1 higher band at 500 base pairs. That means that both chromosomes have an insert. And this would, according to our hypothesis, be correlated to hypersocial behavior. That dog should be super hypersocial. And then there could be a possibility where the dog has 1 chromosome that has no inserted DNA and 1 chromosome that has inserted DNA. So you can see 2 bands in that 3rd lane, and we call that heterozygous. So this is an example of what the students would get and how they would analyze it. They would make a genotype call. And then on the left, I have some sort of crude data of just one gene location. And this is several students. You can see their dog's name. We also assign barcode numbers so that we can keep track of the data and also give it back to Neogen. And then the base or the genotype calls are kind of checked off, and their hypersociability score. So you can graph a class data and see if the trend follows the hypothesis. But if you notice here, one of the issues that you have with just one class, if you don't have enough students, is some of the data may not work. That's what the not available is. And then you may not get dogs with all of the genotypes. So this really isn't a good sample set to get reliable data. So the one thing that we need from you guys is, if you're interested, we would love for you to try this project out. We'll be disseminating all of the curriculum in July at High Tech, and then enter your data into our database so that we can have lots of dog samples and so that the data is more reliable. Okay, so thank you for joining us and listening to our talk. I think we're going to take questions at the end, but if you have any further questions, here's our contact information, and feel free to reach out to any one of us. So thank you.
Well, thank you. That was a great talk. It's really exciting. I'm sure there's going to be a lot of questions, and I do want to mention that feel free to enter questions in the chat, and I will read them off when we start the question time. Thanks again. And now I'm happy to introduce Dr. Elizabeth Boedecker from St. Louis Community College in Missouri, who's going to be talking about genome editing and some of the challenges with delivering that curriculum on genome editing. Thanks, Betsy.
Thank you, Sandy. I'm not a PhD. That is a common mistake people make. So I'm not Dr. Elizabeth Boedecker. I'm just Betsy Boedecker. I am going to talk about our genome editing. The title of my talk is Genome Editing Coursework Delivery in a Post-Pandemic Enrollment Reality. My co-PI was not able to join us this afternoon. She's having some major car issues. And so I'll just be rolling through everything. I'm just going to say to Tracy, Gabby, and her little team, Misty too, I'm going to totally be implementing this, by the way. And I have 4 dogs at home, and I could send you their stuff right now. And so I'm going to just talk a little bit about, first of all, we have a very successful biotechnology program that was established in the late 1990s. It actually has that, bachelor's, that if you have a bachelor's but you don't have any lab skills, some of those folks come for like a 1-year certification in biotech. It is a research and development based curriculum, and so we do provide a little bit of context in terms of a lecture, but 80% of what we do is hands-on, and it's in the lab. And so we are very much a very technical skills training program, and we're very successful. We do what we do really, really well, but we're a small group. I mean, so a big class for us is going to be 12 students, because that hands-on component can be really difficult to manage if you have more than that. And I mean, just about me, I am the district director of our Center for Plant and Life Sciences here. We are a small satellite campus that is embedded in an industry building. So we actually have a lot of our industry partners that come in and hang out in our labs and use some of our several $1 million of equipment that we like to train our students on, because we want them to be trained on what they're going to actually see when they get a job out in the industry here. So I also teach in the program. I'm an adjunct faculty member, so I've been teaching in the program since 2004. We have, just a lot of different things that are, it seems like, always going on. And I have to say, this year has been really crazy so far. And it's only February, so I don't know what my mental health is going to look like towards the end of the year if it keeps going like this. So we have a really high placement rate of our students. They get jobs in the life sciences and they stay employed long term, and we're really, really pleased with that statistic. And we do have external funding. I work very hard to get external funding for our student interns. so they can earn while they learn. So the final piece of their associate's degree is actually placement in an industry lab where they work for a minimum of 150 hours. We have a very tight-knit startup community here, and so a lot of our students get placed with those startup companies, and about a third of the time they end up getting a job offer at the end of their internship. And so we're really pleased with a lot of those relationships that we have in place. And so that's really how our project came to be, is that because of this close knit relationship that we have with a lot of our industry in the region, they had come to us a few years back and were basically indicating they're doing a lot of genome editing now, and they really wish that our students could have some genome editing experience. And so part of our program includes a class title of advanced topics, but it actually encompasses a number of different subjects, things like quantitative PCR, proteomics, just some very specific technologies that our students can then specialize a little bit. And so we kind of felt that this was a great place to kind of plug in some genome editing classes. And so our plan originally was to develop genome editing 1 and 2. We built in a ton of funding for genome editing specific internships. And we actually received this award right at the end of 2019. So yay, right before the pandemic hit. And so we were well along our ways. We had lots of industry discussions on what they thought was going to be relevant curriculum to include, pieces that they thought were really important that we needed to have within those 2 courses. And so we were well along the curriculum development. And then the pandemic hit, and things kind of went sideways on us here. I agree with the previous speakers too, that the industry involvement is so important. Listening to what your advisory board is telling you is, so important to have success in your biotechnology program and in your coursework that you're developing. And so I always want to hit that point. Part of what our original plan included also was some sort of genome editing education conference or workshop. I'd love to be presenting some of this material at High Tech at some point. We were going to do it in person here in St. Louis. There has also been a lot of discussion about doing it virtually at this point, just because it seems like there is a place for virtual conferences, as we're right at one right now, where we're having a nice little sharing of information. So that is still, we're considering what we're going to do with that. And so our funding began in 2019, and we're already on our no cost extension for this year. And I feel we're not super far down in our checklist of things that we were hoping to accomplish. We have offered our genome editing 1 time, and we would like to offer it at least one more time before we solidify what's going to go into genome editing 2. We have our laundry list of what the curriculum needs to include. We just need to figure out how much of the experiments are going to be successful in genome editing 1. And I feel like 1 offering with 4 students is probably not a good enough judge of that. And so we really want to kind of offer it at least one more time, hopefully 2 more times. And then we'll have a better idea of how to kind of pack in what remains into genome editing 2. I have major concerns with ever depleting the genome editing specific internship funding, which is considerable. And that is probably the largest chunk of material that remains in our current grant. And I have met with our program officer at NSF to kind of discuss some of these concerns of mine. And so we're behind for obvious reasons, I think. Low enrollment issues are plaguing us as a college, but it's also a statewide and nationwide issue. We are not alone in this enrollment problem. Even the community college is not alone. even in four-year institutions have seen an incredible decline in the student population that's actively enrolling in classes. And so that is, obviously of great concern. And as an organization, we're doing everything that we can think of to try and boost those enrollment numbers. I also think part of it is that our employment environment is very interesting right now, because I think a lot of people can get a job without any kind of education. And I know that's not going to last forever. We're still going to be here at the end of whatever is going on. But I think that that's also contributing, because they're not necessarily seeing the value in getting education when they can get a job now without any. So I do expect that at some point to be turning around. And we are already, I actually feel like this is the first semester in a number of years where I feel really good about the enrollment that we're seeing is definitely increasing. And so we have students that are coming back now that kind of were midstream in the pandemic, and then they just checked out for a while because they just couldn't handle any more virtual classes, which I completely understand, actually. So these low enrollment issues, I mean, they're obviously a great concern, but I think it's especially dramatic when you are already a very small program to begin with, that impact is felt a lot more severely, because we end up with, 2 or 3 students in a class, and, just for logistical economic reasons, those are not going to be permitted to go forward. And so a lot of canceled classes, and this has ended up really impacting our project timeline in a pretty significant way. So when we finally were able to offer this class, we initially had it on the schedule for spring of last year, but the enrollment was so low, it ended up getting canceled. We actually ended up moving it to the summer session, and there it was allowed to proceed. I think it only had 4 students though, but it actually went pretty well. Summer session is a little bit shorter than our typical semester. So that's another consideration that we want to think about in terms of delivery of the genome editing courses. I also think it is it's complicated material, and so I think having that shorter timeline makes it a little more challenging for students to really grasp what we're teaching them. And so it was a good course overall. It was reviewed well by the students that were participating, but I really do feel like we need to offer it at least one more time before we can really firm up what pieces are going to be going into genome editing too. It was on the schedule, it's still on there as of today, but I do know that this is going to be canceled for this semester. And so and I just put the little blib up there so you guys can see, we have no enrollment for it. Although I will say I'm feeling much more optimistic about it because I know that there's students in my current Biotechnology 1 class that are very interested in taking it. They couldn't take it this semester because it meets on the same nights. So there's no way they can take my class and that class. And so there's still some scheduling. We're doing a lot of, our courses are being offered a lot in the evening. And so there is some conflicts there because they have prerequisite courses that they have to complete first before they're going to be permitted to take something that's an advanced topic. And so it is going to be canceled for the second half of the spring semester, but I've talked to the instructors. I've actually talked to some students too. And so while we are going to be canceling it due to low enrollment for this semester, we are moving it to the fall semester. It looks like it's going to be a 1 night a week class. And I know that there's Biotech 1 and 2 students in classes right now that are very interested in taking that. So I believe we're going to have a pretty good enrollment situation for fall. And that actually makes me really happy. And so I think it's going to go really well. I do also want to mention that we have 2 industry folks that are teaching this class. So we have a senior scientist from Bayer, who's one of our big local companies. And then we have Susanna is the other instructor. They're going to co-instruct it together. She's actually at a small company called CTC, which is a Brazilian agricultural company that actually is in our building where we're located currently. And so I love having these industry folks that are coming in to teach it for us. They're actively doing genome editing in their daily work. And so I think they can add a lot of depth and value to what we're trying to include in the curriculum. And so stay tuned on that. Let's hope everything goes well. So our current plan then obviously has dramatically shifted from that first slide you saw a few back. And so I have already discussed with our program officer at NSF, we're going to ask for another extension. We might be asking for 2 additional extensions because I just don't, things are just not moving quickly enough right now for us to really achieve the overall goals of what we wanted to hit with the genome editing NSF grant. So additionally, we want to reallocate some of that budget because even with 2 more years, the amount of money that we had set aside for genome editing specific internships, even if we filled the classes, I don't see how we're ever going to spend that. And so there was some discussion about reallocating some of that. One of the things that I was very excited to do, and basically I need to just submit my budget and my request, is that we want to take some of that internship money that we had set into that grant and move it into some stipends and supplies. And we want to make at least a couple of days of our teachers workshops that we do every week or every year. We do a week-long teachers training. I'm sure very similar to what Gabby and her team do, where we bring in some high school teachers. Sometimes there's some middle school teachers rolled in there as well. And we want to have at least a couple of days that are focused on genome editing. And so we want to have some very accessible at the high school level genome editing experiments that they can do. I would love to maybe incorporate some of the doggy stuff too, because I think that would be really fun for our teachers to include, and probably pretty easy for them to roll into what they're already doing in the lab trainings that they're providing at a high school level. So we're going to be using some of those funds to try and get some teachers involved in some genome editing that they can get their students involved. So I'm excited about trying to come up with what that will look like. We are what we include in the coursework right now is we definitely want our students, they already have a pretty good basis for what that central dogma looks like and that transcription translation process, but how to think about genome editing as a tool that they can use in modifying organisms. Our industry partners have indicated to us, from the very beginning, talking about the historical perspectives here and how this kind of evolved over time, they think is a really important part of inclusion within the coursework. And so we do talk about those zinc finger nucleases. We talk about the Talon set of experiments and how that all kind of evolved into the CRISPR-Cas9 that everybody is so hot about these days. And so obviously our coursework is including a lot of laboratory work, but we also want to include other things. Again, our industry has indicated to us that it is really important because Jennifer Doudna and her team have made it important that we want everyone to understand what genome editing is. And part of that includes reading some books, reading scientific papers, and having discussions that are really relevant to those topics. And I think having these ethical discussions, even at this very entry level, I think is really going to be a very positive impact overall on the technology as it gets out there, because I think we're all part of the team that we want to make sure the community understands what genome editing is, how it works, what we want to use it for as a society. I think those are all really important pieces that need to be, open for discussion. And I think the more we can get our students talking about it, even in a classroom setting, I think the better advocates they're going to be for this technology out there in the community when they're not working on it. And so that is a really big part of the class too, is basically getting them used to those discussions, even ethical in nature. The laboratory activities involve multiple organisms because, again, we have a lot of agriculture research that goes on, especially right out here where we are located right next door to the Danforth Plant Science Center. But in St. Louis, it's not just about plants. There's a lot of mammalian-based research that goes on, and we have some, my personal thing is I love the stupid worms. I love the C. elegans. And so anytime I can figure out a way to do some C. elegans experimentation, I'm going to try and implement that. So we aren't just focused on plants alone. There is a lot of plant work. There is, both of our instructors work on plants. And so there's a lot of agrobacterium or protoplast work on how to implement some genome editing into plants. But we also have some cell culture based work that we're doing, as well as we're trying to manipulate some C. elegans. We actually might need a couple of small pieces of equipment in order to get that going as efficiently as we would like. And so we're looking at all of that to try and kind of implement it as effectively as possible. And I'm hoping some of those are going to be going into the teacher's workshop for this summer. Genome editing too is really where we're going to, I think, have a lot more of that bioinformatics piece. There is some of that is built into Genome Editing 1. When you're designing your guide RNAs, it has to be part of the discussion there. I'm a big fan of some of the resources that are available. Sorry, Sandy, I'm going to call you out on the Digital World Bio and the Innovate ATE site, because we actually have a bank of iPads that we purchased that have the Digital World Bio software on there, because I think it's really fun for the students to be able to manipulate those different molecules and look at how they fit together, especially when you're talking about a CRISPR-Cas9 system. If they can look at it, I'm a very visual learner myself. And so being able to play with that molecule and rotate it around and see how everything is interacting, I think is a very cool piece of something that we can have included in this coursework. And so discussions about how we get the stuff into the cells, into the plants, into the worms, looking at demos on a gene gun. That was one of the original pieces that we were actually going to purchase, a gene gun here at the college. And in the time from when we actually got the award to when we're actually sitting down with our industry partners, like 3 of the companies had already bought a gene gun, and they're like, you can come demo on our gene gun. You don't need to buy one. And so we kind of already had changed just in the couple of months from when we got the award to when we started planning the curriculum and purchasing equipment. So everything is just, evolving as the project continues. And so how to evaluate those edits? I don't know why I was thinking about that Seinfeld episode where he's talking about, well, you know how to take the reservation, but you really don't understand the most important part is how to hold the reservation. And I kind of feel like the most important part of the genome editing is how do you actually evaluate your edits that you've made? And I think that's going to be a huge part of genome editing too. But we actually have to have some organisms that we've edited in order for that to be taking place. And so there's major stuff that we're still developing for this, but I feel like it's actually making progress now, and that makes me very optimistic that things are going to go well. And I just know that we need to get through that Genome Editing 1 course offering at least one more time before we can really solidify that Genome Editing 2. Happy to share all this information. And so our plans from the first day have been to put this all in a course in a box. Once we have our protocols thoroughly tested and we have our presentations that have really been refined, we are going to be shipping that off to Sandy to put as a course in the box on the Innovate Bio website. So know that will be coming at some point. We're still developing what we want to share with the teachers this summer. If it's going to be a 1 or 2 day, if it might even be the whole week, we don't know yet, but that is all under development. And as soon as we have that really much more refined on what we're going to be doing, I'm absolutely happy to share that as well. Always happy to discuss and answer questions. if I know the answer. I don't always know the answer, but we're excited about how things are moving along now. And so I want to thank everybody for this opportunity to kind of share our progress or lack of progress or hope for progress at some point. And feel free to contact me. My email address is here. Sandy knows how to get a hold of me. And I am absolutely happy to field any questions at this point, and hear the answer to questions that are going to go to the other team that presented to you. So thank you very much for this opportunity. I really appreciate sharing what we were hoping to accomplish.
Well, thanks, both of you. I mean, all of you. These are great. And I actually, this is something, Betsy, that you talked about that you don't always hear. How do people deal with some of the mechanics of actually running their ATE project? And what do they do when things are not going 100% the way they expected? I mean, nobody expects a pandemic, really,
No.
but just the enrollment questions and how you deal with extra money. And I hope you're thinking too about a supplement as a possibility.
Yeah, I think Celeste Carter, luckily right now, is our program officer, and I just adore her. And so she's met with me and has been just incredibly helpful in how we can. I just had, this is the first grant I've ever been a PI on. I've been a co-PI on a bunch of them, but being responsible for all the money and the reports, I'm like, oh. So she has been really, really helpful in just guiding me on what is allowed, what can I do, how can I reallocate those funds. And so I really appreciate that, because it's very daunting, when you're 4 years into a project and you're nowhere near the end. I'm like, I don't know, do we just give you the money back? What do we do?
You start to memorize the PPA-PG, right?
Yeah.
But by the way, that is the online. It's a big book for what you can spend and how you spend it and things like that. And those of you who are going to be applying or working on ATE grants should think about learning, visiting it from time to time. I'm going to just share one request here in the chat from Scott Ryan, who said he hopes that the summer high school training might be virtual or have some kind of hybrid component. So if there is any possibility to share videos, let us know, because we can always post them on our YouTube channel and
great. Share
them out with people. But I know it's easier said than done. And I'm typed PPA-PG in the chat. You can look it up. So questions. John, you look like you have a question.
I won't take up everybody's time, but I just want to say that, hi, Betsy. I also have a gene editing grant, ATE grant, and projects that are involved with having dissemination of course material for our students, as well as for other interested audiences like community college faculty, and we've run workshops under that. And I would love to talk further about your projects and maybe share some of the materials that have been developed for your courses, kind of how we do it. And I think that listening to that, there are some synergistic activities I think that we could have in kind of pulling all those together. So I thought both talks were excellent. So I'm sorry to jump in with that, but I just want to say hi.
I would love to talk to you.
Sounds great.
Oh, and John, if you want to give a talk, email me.
Yes, I'll follow up with that too. Sure. Yep.
Because we'd love to have you tell us more about the kits that you've developed and your, all the things that you've been doing.
No, I appreciate that very much. And a couple of people on here I know that have participated in those workshops in the past, and hopefully would have positive things to say. But I do think that, part of our aim in our project is obviously to disseminate interest and curriculum related to it, because of the importance and everything that you have going on at a specific level. I think, like I said, fruit for good conversation. So I'll follow up with you off to the side, but I just wanted to say hi. And I love dogs too. So not to diminish anything for the other ones.
Yeah, it's great to see you. And I was wondering if I bring, is there a way we could bring like saliva samples and give them to you guys at High Tech, Misty?
Yeah, sure.
Or maybe
we have some extra dogs on backup.
I have dogs, yeah. I have access to dogs.
Yeah,
They're quiet right now, but there are 2 of them here.
that's good.
Maybe we could pick up the swabs at high tech. Maybe that would be easier.
Yeah, picking up the swabs at high tech and then you could send them to us. That would probably be easier.
That would be great. So more questions, please. I can come up with some.
I have questions for the first talk ladies, the dog ladies. And so I am 100% serious in that I'm very fortunate because we have a dedicated outreach specialist, and she is always looking for stuff to do with high school groups, and she's going to be spearheading a lot of the efforts for the summer workshop this year with the teachers. But I would love, the protocols or, what primer sequence, whatever it is that you have that you're willing to share, so that we could start, implementing this with some school groups. And I have a foster corgi. I work with a corgi foster group. I could get you lots of corgi swabs, and I have 3 dogs myself. And so I'm totally going to do this for my dogs.
Yes, there you go. Yeah, so I think probably at high tech is probably our good goal to have all that stuff ready to share out. We'll also be providing it to Innovate Bio, so then you'll be able to access all that stuff there. Yeah.
Well, that's great. We're looking forward to it. Haven't set up my lab at home yet, but I do have dogs like Betsy.
Yeah.
Are there other questions? Oh, I'm sure people must have some. Okay, here's a question. So the phenotypes, when you score the hypersocial versus the non-hypersocial, what are you looking for? How do you tell?
So the behaviors have to do with like how much your dog wants to be in proximity to you. Like, so I know our dog, if you go sit down in a room and your dog isn't there, then the dog comes and finds you. So that sort of thing would be one of the behaviors. And then if your dog gets anxious when you leave, that's another big thing. And so we are just focusing specifically on the hypersocial behavior, and so the questions are all centered kind of around those 2 specific things. There are some additional questions that have to do with that were in the original paper that had to do with if your dog gets distracted when other people are around. Like, does your dog just focus on you, or is it easily distracted? If it's easily distracted, it'd be more hypersocial. And what was the other one? Attention bias. Yeah, and then if your dog is good at interacting with strangers. So is your dog just attached to you, or is it attached to all people?
Like labs.
Yeah, and it does. Yeah, different breeds tend to have different behaviors, and so as we're collecting data, we're hoping to add more of that breed data and like the age of the dog, so that we can have more data to work with and analyze.
Can you tell us anything about the mechanism? I mean, what are the genes?
Yeah, so actually that is a really interesting aspect. Is the gene is associated with a human disorder where there's a big deletion on chromosome 7. Like, Gabby, do you remember the name of the disorder? It's WBS is the abbreviation. But the people that have that disorder, they tend to be super hypersocial. So that big deletion on chromosome 7. And then that is how then the people found at Princeton and Oregon State found this spot on chromosome 6, since it had was homologous then to what was found in the humans.
Oh, that's really interesting. And we have a request. If you could share the link to the paper in the chat, would be great.
Yeah.
I think I remember reading about that disorder. I feel like it's like Williams syndrome or something.
Yeah, Williams, and then something that starts with a B, syndrome. Yeah, and there are some other like facial structure is different in the people as well. It's not just the hypersocial behavior. So, but that human component, yeah, and that's one thing that we're hoping to kind of wrap into the final curriculum as well.
That's interesting. Do dogs have that different facial structure?
I don't think I haven't read anything about that.
I'm just wondering.
Yeah.
You're getting a lot of thanks especially about Betsy, for you being so open about the challenges that you've had and how you're working with your program officer to address those and get more time to complete the things that you really feel are important about your project. I think people sometimes are afraid of their program officers and they don't realize that program officers are interested in your success and want to see you do well. So, okay, I want to remind everybody about the survey. I'm going to put that in the chat one more time. And I hope we see all of you next week when we have Chandra Arora from LA Mission College talking about project-based learning with adults and recruiting adults into her program. And we have Susan Ingersoll, who's here talking about project, okay, I'm going to forget the name. I feel like it should be like Hot Start PCR or something. Susan, tell me again.
So it's about improving the biotechnology workforce by improving their internship experiences.
And Susan gave a great talk at High Tech last summer. So I'm sure we're going to learn a lot and hear a lot of interesting things next week.
Thank you.
And any other questions from people? All right. Well, I'm going to let all of you do the survey then, and we'll hope to see you later.
Thank you.
And thanks to our speakers. Sorry, I forgot that. We really appreciate you coming and talking about what you're doing. It's so much more interesting than trying to read the abstracts, trying to figure out what are they doing.
For
Also,
sure.
it's alive, right? It's not what you were thinking about when you wrote the grant. It's what you've actually found and you're working on.
Yep, for sure.
Which is really great. Okay, well, I'm going to sign off.
Okay, see you later.
See you all later.